Hello, everybody. This is the review for the second exam. I anticipate the second exam will be on either the Wednesday of next week uh, or on the whatever that's going to be the first or the third, which would be the Friday. We'll decide tomorrow in class, which will be obviously this coming Wednesday. So allow me to go from there. So there are three chapters involved from this. One we have not completed, which is the viral chapter, though it will not take long to complete it. We'll have that done probably tomorrow, latest Friday. And it starts with chapter 5. Uh, Let me rephrase. It starts with a chapter on disinfectant. The chapter numbers are different. So chapter 5 was Nestor, and chapter 9, I know it's a bit confusing, is the Cowan textbook, but in either way, the material is the same. So it starts with disinfectants, germicides, physical and chemical methods of control, etc. And my first series of questions involves categories of agents that are used. And so you will not get the name of specific agent. I'm not going to say orthopaldehyde or glutaraldehyde. I will say aldehydes. I'm not going to say it's either biguanides or chlorhexidine. And they're effectively a category unto themselves. Phenolics, halogens could be chlorine or iodine, for instance. So look at the category, not the name of the specific item that's there. So understand that some items are sterilants, and even in a liquid sense, those chemicals can be sterilants, the way that gaseous agents can be sterilants as well. So if they're sterilants, then we can use they'll effectively be sporicidal under the right conditions. Why do we use one than the other? Because it's an alternative to using heat. So, heat sensitive items, or certain chemical sensitive items, or moisture sensitive items, or items that you're concerned about being dulled or having a residue on them, all of those are potential problems. The most commonly used agent from a liquid perspective are the aldehydes by far. Typically, glutaraldehyde, which is the one that's been used most commonly. I used it myself in the office for many, many, many years, and that was there. And it's available agent. I'm still confident use today. When they talk about antiseptics, those are ones that are by definition not sterilants. And so we have two of them that we utilize a lot in surgery, both as prep agents and agents that we use for scrubbing hands in surgery. And so those agents are either in the chlorhexidine biguanide category or the halogen category. They both work the same, and they're effectively betadine is enhanced by soap, and chlorhexidine products are enhanced by soap. So they're a wonderful antiseptic agent. And because of being combined with the soap, they're not quite as concentrated, so they're a lot kinder to tissues. So we can use even chlorhexidines on indwelling catheters and things like that, and betadine soaps and betadine products that have been diluted are all over the place. So they're very common. How do you tell the difference? Betadine basically is a brownish kind of soap. Chlorhexidine typically is a reddish kind of soap, reddish or pink kind of a soap. So think of the soaps as being white mixed with a red compound, you get pink. The betadine, which is more brown, so it's kind of a golden brown or gold, like a yellowish kind of a color. And that's how you distinguish between the two of them. We use ethylene oxide, a poison gas, all the time because it's great for paper items. It's great for items that you can't expose to moisture or expose to heat. And so it penetrates very, very well. So every time you get a sterile Q-tip out, like you do in the lab, or a sterile pipette that you do in the lab, a needle, a syringe, all of those things that you use commonly, not in everyday use. Even if you go to the grocery store or the drugstore and you buy a pack of sterile gauze pads, just a couple of, just a couple of pads each, those are typically sterilized with gas that are used. Things like the only time we really see a lot of radiation in common usage, other than some in the food service industry for sterilization, is probably the sterile surgical blades, because it's really a very effective means of treating them, and it doesn't dull surgical blades. They're certainly the ones I've seen the most that are there. So that's the first series of that. The two that are used most commonly by far and away are the halogen categories. The disinfectant agent is bleach. Okay, it's used all the time. It's corrosive. It's toxic. It's got a nasty odor. It's not something that you would use routinely. We tend to really. There's probably a While it's a very, very useful agent, it's just something that has such a distinctive odor. It's very, very noticeable, and certainly can't be useful for people with respiratory disorders. But it's tremendously inexpensive. I mean, you can get a gallon for $3 or $4 at the store, and you can dilute that, and it's still very, very efficient. And that remember that when you're dealing with cold liquid sterilizations, it's a matter of time. So that wiping down something with bleach, diluted or concentrated, is a really great disinfectant. It's not sterilizing a tabletop. You'd have to have that on the surface for a minimum of 3 hours, and that's just not feasible. Plus, it would typically damage the surface. For instance, you can't use bleaches on stainless steel tabletops. That's a big mistake because it'll tarnish it, and then the value of stainless steel is lost almost immediately with exposure to bleach. If you didn't know that, you should be aware of that. For anything of stainless steel at home, be careful with bleach that are there. So what made iodines more efficient, because they were irritating to skin and to tissue, was combining them with a protein carrier called povidone. So they made povidone and iodine together, and it became betadine, and it was a lot less skin irritation and tremendously useful with soaps. And again, that's still a halogen category. And what did we use prior to these? In the early days, iodine was around, but we used a lot of metallic compounds, particularly gold and silver and dental fillings, and mercury. Okay, mercuric compounds, mercuric vapors, and things like that. We found out that they're a neurotoxin, and that's the big concern about thimerosal being used very, very commonly, as well as formaldehyde, for instance, as a preservative in certain immunizations. And that's the concern with that is certainly with the relatively underdeveloped nervous system. Understand that things like the metallic agents, like mercuric compounds and things like that, or tincture of merthiolate, which was the one that was most commonly used in the past, are something you just don't see a lot of them anymore. But you and I ingest probably more mercury if we're coming on the season for Lenten fish fries because there's plenty of mercury in a lot of fish. But when you're exposing an immature nervous system to it, there's a big concern. So mercuric compounds, it's the neurotoxicity, which is their big problem. So what's another valuable agent? Peroxygens, because they release toxic levels of oxygen gas. And I mean, well, it's oxygen. How can it be toxic? But understand that organisms don't have the means to deal with the toxic derivatives of oxygen that occur when they're exposed to oxygen. That's why using hydrogen peroxide is effective. That's why we utilize it for wound care. That's why it's environmentally friendly. It dissipates because it changes to water and oxygen gas, so it doesn't linger on things like that. And even at very high levels, it's a sterilant. So that's sort of a synopsis of the common chemicals, and then we deal with the flip side of that. And let me just check something here, everybody, if you don't mind. I want to make sure because I have to do this on a different computer today, and I don't want to end up. So I've got to make sure by checking my settings whether or not this call could drop, and I don't want my call to drop whatsoever. Good, it will not. So now pardon me for that delay in that. I left my power cord for my work computer at the office, and I'm at home dictating this. Now, physical methods of control: heat, radiation, filtration, mechanical activity. Those are physical methods of control that are there. And yes, they can achieve sterilization. Remember, sterilization, we don't put prions into the discussion, even though we feel that they're harder to remove from the environment than endospores, but it's the definition of removal of all microorganisms and viruses including bacterial endospores. If we got the endospores, then we assume it's sterile. And again, when we know more about prions and how to control them and what their impact is on daily life or in surgery or in anything that we have to have sterile, we'll know better about that. The distinction between disinfectants and antiseptics. Antiseptics on skin, disinfectants on surface. Okay. And so we see those types of, that's why I consider hand washing to be more mechanical. And I don't know whether you'll do the lab in your micro lab, the hand washing lab, where we use no soap and we use Purell and we use bar soap and different kinds of soaps, to see that there really is not a tremendous distinction in the effectiveness of hand washing. If you do it for about 30 seconds, you're good. So I think it's more about mechanical removal. You should also remember, when we're doing, and we use the autoclave for, which is basically pressurized steam or superheated steam. The numbers are this: 121 degrees Celsius for 15 or 20 minutes. I think I use 15 on the test for, okay. So once you have that 121 degrees Celsius for 15 minutes at a pressure of 15 pounds per square inch, which is typical pressure, then you're going to ensure sterilization. Personally, I've always think that longer is better, and having it for another 15 minutes isn't the worst thing, which we did in the office. We did it for 30 minutes just to be sure. Sterilization is a biological process. You're destroying endospores. You have to see, in order to verify that it's sterilized, you have to be able to monitor it. So there's a means that is used in hospitals and in large medical facilities that dispense sterile goods and things like that. Let's say Johnson and Johnson, with all the sterile materials that you might buy at a local pharmacy or at a grocery store. And what they do is all they can do is certify that their machines are sterilizing, and that's done by inserting an endospore containing packet and making sure it's destroyed. So there's endospores in the tube. Then you have to make sure. Then basically it takes 24 to 48 hours to incubate that tube, which has an indicator dye to make sure that the endospores are not being able to flourish or to germinate. That's the interesting part. All those tapes that you see, those are just heat sensitive materials. So autoclave tape or autoclave indicator strips will tell you that the heat got there. That's all well and good. And I mean, I would be fairly confident that there'd be no problem. We used that in the office. We did not use the endospore containing tubes, because that's something, when you're under governmental controls or Joint Commission activities, these governing bodies, then you have to keep more scrupulous records. But most doctors' offices, you just make sure that you maintain it properly, and you're probably going to be fine with it. But that's really the formal way of knowing it. So again, that's the distinction. You can still sterilize with liquids. It's a matter of time, and it's not minutes, it's hours. Typically, anywhere between 3 to 12. The average time would be about 6 to 10 hours to sterilize. So and again, when you're looking at disinfection, when you hear about pasteurization, we always talk about foods that are pasteurized and consumables. It's still a form of disinfection. Roughly 63 degrees Celsius for 30 minutes. That's the batch method. You can increase the temperature another 10 degrees Celsius and decrease the time. That's the high temperature method, which is used a lot because time is so important in large amounts of pasteurization in the food industry, and even for higher fatty substances, let's say ice cream, sour cream, that type of stuff. You're going to have to even increase it another 10 degrees Celsius, and maybe for 15 or 20 seconds again. So those are important. Again, hydrogen peroxide under the right conditions, which is vapor phase, superheated, can be a sterilant as well. The beauty of these, and understand the advantage we have with a lot of these things. We use alcohol because it evaporates quickly, but that's the problem with it. It evaporates quickly. It doesn't leave a residue. So it's useful because it's a dry system and you're not walking around with wet hands all the time. That's why all the hand sanitizers have been just tremendously useful, and you see people using them all the time, almost to excess. And I think you have to be careful with that as well, because of the problems with phenol and phenolic compounds, we switched a lot to quaternary ammonium salts. The phenols tended to accumulate in tissues. There have been problems with them, so they switched from quaternary, I beg your pardon, ammonium salts away from things like triclosan. So that's the distinction. The next section, even though there'll be a few more questions on the germicides and disinfectant area of it, that are really about the viruses. And I think the bulk of the test is the viral thing. So how do we classify them? I went through this in class. We talk about whether they're genome, whether they are an enveloped or non-enveloped, which they sometimes call naked viruses, and their architecture, their size, their shape, whether or not they're isometric or helical effectively. Viruses are everywhere and they can affect all living things, all domains. So they can affect eukarya and prokaryotic domains as well, which means archaea and bacteria. And they're all significant. So you have to know it's less about the diseases and more about the structure. So certain things that you're familiar about, we know HPV causes genital warts as well as other kinds of warts. Okay. We know that viruses are effectively a genetic material, DNA or RNA, never both, once again, inside of a protein shell we call a capsid. It's the shape and the arrangement of the capsid, pardon me, that distinguishes what kind of virus it is. That being said, so that's the distinction between the icosahedral or the filamentous or helical viruses, and we call that combination the nucleocapsid that are there. And protein spikes that extend either from the capsid or from the envelope, and that's the big distinction why enveloped viruses are considerably easier to control in the environment with disinfectants because we can target the fatty envelope, and there's a lot of solvents and a lot of materials that are in the disinfectant antiseptic category that can clobber the envelope. But those protein spikes are where it attaches, and that is on specific receptors, and that establishes the range of the virus, what species or what target cells it can attach to. So the distinction having to look at when we talk about icosahedral or isometric, it's typically the 20 capsomere, which are the individual subunits arranged in this basically an equilateral triangle, and becomes effectively the equivalent of like a 20-sided die in a board game that are there. The big distinction for me, if you wanted to look at it in a broad range, is DNA viruses are more often latent, where RNA viruses have a greater tendency to mutate. We're going to explore that even more significantly in lecture over the next day or two when we look at both the coronavirus and perhaps more relevant, the influenza virus, because it's a more common problem that we face that are there. The oddball virus of them all that really impacts us is HIV, which is considered a retro or a backwards virus. It goes, rather than being RNA or DNA, it's RNA that becomes DNA. And that really is interesting. I always classify it as the worst of both worlds. It can become latent and become a permanent dwelling in a cell line, which it does with a helper T cell, the single most important cell in your immune system on one side. And then it can mutate very quickly like an RNA virus. So it has the worst characteristics of both of those. You should be familiar with how viruses are spread. And that has to do with, and these are the common ways that they spread. It's not the exclusive way. So and basically it's a matching question, and the key is fecal to oral, respiratory, zoonotic, or sexually transmitted blood or body fluids. Those are the four categories. So the common cold, hey, respiratory, you know that. But the other ones are distinctive. Hepatitis, we have two different types. Hepatitis A, fecal to oral. Hepatitis C, and as well as hepatitis B, sexually transmitted blood or body fluids. Yellow fever, zoonotic. Zika, zoonotic. Rabies, zoonotic, for instance. Where measles, even though it shows up on your skin, chickenpox, even though it shows up on your skin, respiratory. Polio, even though it affects your nervous system, isn't, and they got this all wrong. They thought it was respiratorily spread. It's fecal to oral, contaminated water, just for examples. So some of these are obvious. You know, cold and respiratory viruses are generally respiratorily spread. Most diarrheal diseases and things like that are spread fecal to oral, for instance. With herpes, typical herpes type 1, herpes type 2, typically sexually transmitted. Things like that. Infectious mononucleosis, close intimate contact, sexually transmitted, whatever, even though it doesn't have to, sexual doesn't imply intercourse, it's close and intimate contact with blood or body fluid. So saliva is a body fluid in that regard. So we're going to learn about the strategy with regard to how these viruses interact. When they go to animal cells, the virus is ingested in totality. So it enters the cell. In bacteria, they just inject the genetic material. In animal cells, the whole virus comes in, including the capsid, and so you have to remove the capsid enzymatically. That's called uncoating. And viruses, very interesting how it's like an assembly line how they replicate. All the components are replicated, then they're assembled, and we will go over this even though we have not done so when I'm doing this dictation. So the categories are important in what they are. For instance, paramyxoviruses, you will see, have 4 diseases that are all respiratory: measles, mumps, parainfluenza, which is like a mild influenza, and this RSV, which is a big player now, respiratory syncytial virus. And viruses either explode out of the cells or they're sort of spit out. Enveloped viruses are kind of spit out. We call it budding or shedding, where a common cold is a lytic virus. So it fills up a respiratory cell, the cell bursts and spreads 500 or 1,000 viruses. Viruses don't make toxins. Viruses, bacteria make toxins. That's how they do most of their damage. Not all, but most. Viruses, most of their damage is indirect, meaning it's from your immune system. They attach to certain cells, and in the process, they damage them. So when you hear about a hemorrhagic virus like Ebola, it's damaging the lining cells, and capillaries begin to burst and leak into organs, and the organs get damaged, and then there's a huge inflammatory response or an immunological response that's part of it. The damage from COVID or influenza is almost essentially all immunologic. And we have in subsequent chapters, we go into this. So know those categories. The pico RNA viruses are a broad category. I mean, you have polio, hepatitis A, rhinoviruses, enteroviruses, Kawasaki viruses. I mean, there's a lot of viruses that are in that particular category. And viruses, the only viruses that don't directly impact us are bacteriophage viruses, because they impact bacteria, but they can make the bacteria more nasty to us, like they make botulism produce a toxin, for instance, or they'll make diphtheria produce a toxin. Those are infections from bacteria that become more deadly to us because they have been undergone viral infection. You're going to see it's something called lysogenic conversion. So we're going to look at the way that viruses impact you. Remember, before they can do any harm to you, they have to attach, then they have to be adsorbed or brought into the cell, then they have to uncoat, then they have to undergo this. They basically take over the cell, replicate the components, assemble the components, and release the components. So there's a lot to it. Viruses are very significant because of their association with cancer. 15 to 30%. I say this number again and again. You can read a lot of authors. This is typically the number, are directly associated to viral infections, and certainly HPV is number one on the hit parade. So understand those and look at all those categories. And on the study guide, you will see that I have given you the key diseases that go into those particular categories. So I have another basically modified matching question, and these are, for instance, I'll give you the answer key. You have to look up the viruses. Rhinovirus, common cold. Herpes virus. Remember, we went over 7 of them. So you've got simplex 1, simplex 2, VZV, Epstein-Barr, cytomegalo, HHV6, HHV8. So know what they're about. Paramyxo, measles, mumps, parainfluenza, RSV. Orthomyxo, strictly influenza. Retro HIV, for instance. Those are the five. That's the answer key. So you got to figure that the diseases I'm looking for are in that category. And the last unit about this is the genetics unit that comes at the end of the test. There's roughly 10 or 15 questions that are there. Why are bacteria such a problem? It's because effectively they're ready to mutate. Remember, our cells are diploid, bacterial cells are haploid. Our cells, the chromosomes are in what are called chromatin. They're not ready to replicate rapidly. Bacterial chromosomes are already supercoiled. Those are like R's that are entering mitosis. The biggest concern has to do with conjugation. That's because of the F plasmid. The F plasmid, at a minimum, has the information to assemble the pilus and to accomplish the transfer of genetic material. If it has resistance, that's just something extra that comes with. But an F plasmid, at a minimum, those two factors: F pilus formation and the ability to conjugate that's there. If it adds resistance or it has resistance, now we can also categorize it as an R plasmid. So an R plasmid can be an F plasmid, or an R plasmid may not be an F plasmid. Okay. And so we have different categories of mutations. Mutations are errors in DNA. They can be spontaneous, just through normal replication. They can be induced with exposure to mutagens like radiation or noxious chemicals. Ultraviolet light can induce mutations, typically thymine dimers. Ionizing radiation, gamma rays or X rays, are the biggest physical mutagens that we have. The different categories of mutation. Silent mutation means even though there's an error in DNA, it doesn't affect the actual protein or polypeptide that's produced. A missense mutation indicates that it's incorporated a wrong amino acid. It doesn't mean it's going to alter its function dramatically. It may or it may not. And a nonsense mutation means that a stop code has been encountered prematurely and the protein or the polypeptide is not assembled properly. Most of these are caught by mismatch repair, but at best it's a 50-50 process, where our DNA polymerase 3 is much more assured of not just correcting a mismatch, but removing the damaged DNA. So when we talk about horizontal or lateral transfer, when we inherit genetic characteristics, it's vertical. Bacteria, that means in the same generation, that they can pick up DNA from another member of their population. They pick it up from a donor and they are the recipient. Transformation and transduction don't require contact. Transformation is naked DNA from ruptured cells that have DNA floating about, and transduction is effectively the same process, but uses a bacteriophage virus as a vector to move it. When a cell is F positive, it has the F pilus, it has the F plasmid, okay? But once it undergoes conjugation, then the recipient cell, because now it has a whole F plasmid, becomes F positive as well. Oh, don't forget about the Ames test, okay? The Ames test is the one that involves basically what's called a Salmonella auxotroph. It's a mutated form of Salmonella that is reverse or backwards mutated. If it's such a powerful mutagen to do that, then it's something we should not be coming in contact with on ourselves or internally. So that's the test that the FDA uses. So it's a reverse mutation of Salmonella. And in order to do that, they have to use some rat liver enzymes because it needs, basically mammalian enzymes in order to activate it to see what effect it would have on us. And so that's what it is. And transposons. Can't forget those. Those are the genes, quote unquote, jumping genes that can move from place to place. McClintock's research, typically they move on a plasmid. When they move, they can insert themselves anywhere or multiple places on a strand of DNA. And that's where you saw that pattern in the corn that was so well known. So that's everything for our review. I will send this to everybody. If you have any questions, feel free to email, ask me in class. We'll set up the date, etc., etc. Thanks again. Bye-bye.